Hi everyone, and thank you for coming. So I'm Lavinia and I'm going to start off the talk and then Dave's going to pick it up a little later on. And I'm going to start by firstly giving you some information about the genetic code. So the problem with giving a genetics talk, we will almost definitely start talking about some technical words that we need to make sure at the beginning everybody understands. So I'll start off with that. I'm then also going to talk about the Human Genome Project, which isn't something that Children of the 90s was part of, but it was a really important finding in genetics that allows us to do the type of research that we now do. I'm then going to go on to talking about the genetic data that we have in Children of the 90s and how we generated that data. Then I'm going to talk about how we find genes that cause certain conditions. So you can see here a headline where scientists have discovered the gene that causes baldness. So this isn't a Children of the 90s paper, but how actually was this carried out? How do we actually find these genes, and what does it mean when you see a headline such as this? At that point, Dave's going to take over and answer the question, why do we do this? Why are we interested in finding these genes? He's then going to give you a specific example where Children of the 90s was really, really instrumental in helping to understand how a particular gene influences obesity. Then lastly, he's going to go through some of the other genes that we've found using Children of the 90s data, genes that influence certain traits and diseases that we've found over the past couple of years. Okay, so let's get started. So here is a diagram of what we call the genetic code. So as you almost all know, I'm sure, your body is made up of millions and millions of cells, and each of these cells contains the genetic code inside a nucleus in the middle of the cell there. And as you might remember from your school biology lessons, the genetic code is contained in chromosomes. You see the chromosomes here that are inside the nucleus. And as humans, we have 23 pairs of these chromosomes. So we inherit one of each pair from our mum and one from our dad, and these make up the 23 pairs that contain all of our genetic code. You might be able to see on this diagram that there's something tightly coiled up inside those chromosomes, and if we actually unravel this, we can see the DNA that's tightly curled up into those chromosomes. So this is a DNA, and it's called a double helix because we can see the two strands that are interwoven around each other. Attached to those strands, if we zoom in a bit further, you can see some letters, and this is what we mean when we're talking about the genetic code. So your genetic code is made up of these letters of A, G, C, and T, and the combination of them throughout your genome is your genetic code. A stretch of these letters together make up a gene, and in this diagram, it's only a few letters long, but actually in reality, a gene will be thousands of letters long. And if one of these letters, say this C here, was to change into a T or any other letter, we call that a mutation. And when there's a mutation in your genetic code, it can actually stop a gene from working, it can make the gene not as effective, or it can change what that gene does. And it's those things that can then go on and influence you in your life. So it can cause a disease, it can cause you to be taller or shorter, it can change what hair colour you have. And so these are the kind of things that we're looking for in Children of the 90s. Okay, so now I'm going to take you back to 2001 when the first human genome was sequenced. And does anyone have any idea how long a human genome sequence is? How many letters make up a human genome? Anyone got a guess? Come on, shout out a random number, someone. 14,000? Higher? Much higher? Not quite. It's 3 billion letters long, which is incredible. So this is what a very tiny part of it looks like. You probably can't see this, but there's a list of A, G, C's and T's that make up a tiny part of this person's human genome. So as I said, it's 3 billion letters long. So what would this look like if we wrote it all out? Well, if this was a small part of one of the pages in a book, the book would actually be hundreds and hundreds of pages long. And not only that, there would be more than 100 books that it would take to write out your entire human genome. And a couple of facts here. So what does 3 billion letters look like? Well, not only would it fill up this entire bookshelf, but if you were to stack the letters one on top of each other, a millimetre apart, your human genome would be 7,000 times the height of the Empire State Building. Or to put it another way, if we were to lay it along the ground and started it here in Bristol, it would actually extend to beyond Athens in Greece. And that amount of information is in every cell of your body, so it really is a huge amount of information. So as I said, the human genome was sequenced in 2001, and that's 11 years ago now. So if we've got the human genome, haven't we finished? Have we found all the information we need? Why are we bothering doing studies in children of the 90s? Has anyone got any ideas? Yeah, so if we go back to have a look at a section of it, if I told you this was a particular person, what can you tell me about that person? Nothing, really. It's just a lot of letters. It's like we've got the instruction manual, but we actually don't know how to read it or how to interpret it. So that's what we're trying to do now. We want to annotate the genome, work out where the genes are, and find out what those genes do. So if you see here, you've got a list of the letters again, A, G, C's and T's, and then this section here has been highlighted in pink, because we now know that that section forms part of a gene called DISC1, and all genes have terrible names like this that are hard to interpret. But you can also see that there's lots of single letters in this code that have been highlighted. And those are where, when we compare different humans, we see differences. So a lot of our DNA is exactly the same between different individuals, but there's some places where we vary, and they're the bits that are important to us. So to zoom in just a little bit on this, at the beginning of DISC1, you can see there's this A position here, and most people will have an A at this position in their DISC1 gene. Some people will have a mutation and they'll have a T at that position. And if you have a T at that position, you're at a slightly increased risk of having schizophrenia. So these are the types of things that we're trying to identify in the genome. The other thing about the Human Genome Project was it sequenced one human genome. And actually what we want to do now is look at the genomes of thousands of individuals, and by comparing them we can work out what different parts of the genome are doing. Okay, so getting on to children of the 90s, how have we actually generated genetic data? So some of you will remember coming to a clinic and having a blood sample taken. That blood sample would have been put in the freezer for several years in our laboratory. Then at some time later, the blood sample containing all the cells in your body would have been taken out, and some DNA would have been extracted from those blood cells. That was probably put back in the freezer in our lab. And then, because the human genome was only sequenced in 2001, so that's 10 years after Children of the 90s started, there needed to be a technology catch-up before we could do genetic studies. And the human genome was sequenced, and there was lots of technology development. And now, many years later, your DNA samples can be taken out of the freezer and put through one of these very complicated, very expensive, but very boring-looking machines, and using one of these chips, which are only a couple of inches tall, and these DNA chips are put in the front of the machine. Your DNA samples are put in that machine. Some very, very complicated information comes out to start with, and eventually this can get turned into some genetic information, the A, G, C's and T's that we saw earlier. So once we had the genetic data from the participants of Children of the 90s, what do we do with it? Well, to take you back to the headline that I showed you on the first slide, where the headline said, "Scientists discover the gene that causes baldness." How was that actually found? What did those scientists do? Well, here's my little diagram to help you understand this. So we get a load of participants that are bald and a load of participants that aren't bald, and we split them into two groups. And then for one particular position in the genome, we look at what letters they have there. So remember, you get one letter from your mum and one from your dad, so everyone has two letters. And you can see here some individuals have inherited a T and an A, some have got 2 A's, and some have got 2 T's. Now, hopefully, you'll be able to see that in the top part, the people that are bald, there are more A letters than in the bottom part of this diagram. So what we say is that if you have the A at this position, you're more likely to be bald. Okay? But what it doesn't mean is that this gene causes baldness, because although you'll see some people here that have 2 of these bald A's, there's some that don't have any of them. This person just has 2 T's. So something else is causing that person to be bald. And equally, there might be some individuals that have 2 of these A's that aren't bald, so it's not causing baldness in that person. It's just a way of saying that this position, a change in this position makes you more likely to be bald than if you didn't have that mutation. The other thing to say here is that this is just looking at one position, and when we do studies in children of the 90s, we actually have information on 2.5 million positions throughout the genome. So what we have to do is we do a statistical test to compare this group and this group, but we have to do that test 2.5 million times. And this, obviously, there's a lot of data, this takes a long time, so we actually have supercomputers in the university that can do these types of calculations. Okay, so that's how we find these genetic associations. Now I'm going to hand you over to Dave, who's going to tell you why we do it and give you some examples.
So to just kind of summarize what Lavinia was saying, for most of the diseases that we're interested in looking at, so these are the common diseases, so diseases that a lot of the population have. For most of them, genes and environment are important, so they're not just genetic diseases, or they're not just caused by the environment, they're caused by a combination of the two. And for most of these diseases, they're influenced not by just 1 or 2 genes, but they're influenced literally by hundreds of genes across the genome. So why are we interested in doing this? Well, we're interested because finding individual genes helps us understand how the disease develops, and if we understand better how the disease develops, then we can better understand how to treat the disease. So I'm just going to talk about one disease in particular, because I've only got about 10 minutes, and that's obesity. And hopefully what I'll show you is that the children of the 90s cohort played an integral part in the discovery of the first gene for obesity. So obesity it's kind of a dangerous thing, okay? If you're very overweight, then in later life, you have a much increased risk of suffering from all sorts of nasty things. So, for example, you're at much higher risk of having a heart attack or a stroke. You're at much greater risk of having some forms of cancer, of type 2 diabetes, which is a particularly nasty disease where your body stops becoming sensitive to sugar and glucose, and a whole range of other nasty conditions. If we look across the Western world, we see something very striking, and that's that the rates of obesity are increasing dramatically. So back in 1980, about 15% of adult Americans were obese, deliberately picking on America here, to nowadays, it's more like a third of American adults. In fact, about 75% of all American adults are either overweight or obese. So this is an increasing problem, and it's brought about because we're eating more sorts of this stuff and we're leading more sedentary lifestyles. How do we know that obesity has a genetic component? Well, what we can do is we can look at twins. So obviously there are identical twins. These are twins that share all their genes in common. They're genetically identical. And we can compare those twins to non-identical twins, which only share about half their genes on average. So they're just like ordinary brothers or sisters. And what we find is that identical twins tend to be more similar in terms of their weight than do non-identical twins. And we can use this fact to estimate how genetic obesity is. And when we do that, we come up with a figure somewhere around 50%. So it's kind of half genetic, half environmental. So this is a diagram illustrating the discovery of the very first genetic variant underlying obesity. And it's in a gene called FTO. So this was found by comparing a whole lot of individuals who were heavier than average to a whole lot of individuals who were of normal weight and comparing their genomes, just like Lavinia was talking about. So what you see here is one of these, we call them genome-wide association analyses. So you can imagine that on this x-axis here is the human genome. So it begins at chromosome 1 all the way up to chromosome 22 and the sex chromosome. On the y-axis is kind of a measure of the strength of signal at that position on the genome. So whenever you have a big tower like here, it indicates that here's a gene that has something to do with obesity. So this little tower here on chromosome 16, the FTO gene, this is the first one that got discovered for obesity. Now, the children of the 90s cohort played a really important role in this discovery. Now, you might remember from coming into clinic many years ago, you might have got measured on a machine which looks a little bit like this. This is called a DEXA machine, and what it does is it produces like an X-ray of your body and kind of divides up how much of your body is fat, how much is muscle, and how much is bone. And what we were able to show in the children of the 90s is that individuals who had the high-risk genotype for FTO had much more fat content in their bodies than people who had the medium or the low-risk genotype at FTO. Whereas when one looked at muscle mass, there wasn't much of a difference between the different genotypes. So FTO is affecting your fat mass specifically. Okay, so we know that a gene's affecting fat mass. The next question we're interested in is, how does it do it? So again, children of the 90s was able to give us some information about what FTO actually does. So you might have remember being mailed some questionnaires to do with your dietary intake. Well, here's how we've used that. Again, we can see that people who have the high-risk FTO genotype have a much higher energy intake than people who have the low or medium-risk genotype. So in other words, people with the high-risk genotype are eating more, essentially. The plot gets a little bit thicker. So again, you might remember that as part of Children of the 90s, we asked you all sorts of questions about how much you exercise. And in fact, some of you might have had to wear a little device called an actigraph, which kind of measured how much you were running about. Well, it turns out that exercise limits the effect of FTO. So if you're an inactive person, if you have the high-risk FTO genotype, then you have a big weight difference in your BMI between having the low or medium-risk genotype. But if you're an active person, then this kind of limits the effect of FTO. So the difference between the genotypes in terms of your BMI is not quite as great if you're an active person as compared to whether you're sedentary. One of the cool things we can do as scientists, just because we can, is we can take a cute little critter like this and we can genetically modify him. So now we can modify him so he doesn't just carry 2 copies of the gene, but he carries 3 or 4 copies of the gene. And when you do that, you find that mice that carry more copies of the gene are heavier, and you find that mice that copy more copies of the gene also eat more. So you're kind of getting a consistent story here. FTO is increasing your body weight, and it's doing it because the person eats more. So that's just one of the many successes in terms of the genetic studies in children of the 90s, but there have been many more. And I've just tried to kind of list some of the key highlights to my mind on the next 2 slides. So we've found genes which contribute to risk of eczema, so that's kind of a skin disease where you get scaly skin. We've found genes that influence bone mineral density and future risk of osteoporosis and brittle bones. We found genes to do with birth weight and lung function. Because there's so much data in children of the 90s, we've also looked at more kind of quirky things. So something called 2D/4D ratio. So that's basically the ratio of the length of your index finger to the ratio of the length of your ring finger, which is something that psychologists get very excited about because it correlates with all sorts of interesting things, all sorts of interesting psychological traits. We found genes to do with head circumference and face shape, and even when you got your first tooth when you were a kid. So lots of interesting genetic discoveries. To kind of put this more in a historical context, this is where the scientific community was in 2001 in terms of knowledge of genes contributing to disease. So this is the 23 chromosomes that humans have, and in 2001 we didn't know of any genes affecting complex diseases. This is where we are now, at the end of 2011 anyway, so that's the same diagram as you saw before, but every time you see a little coloured dot, it means that we know about a gene that causes a disease or a trait. So at this stage, we're up to about 1,500, which is quite an achievement in the last 10 years. So I'll finish up now, and we might open the floor to questions. Thank you.